So if you go under PowerPoints here, you can go down to special senses, and then, like I said, there's a great documentary and on the secrets of the mind, part of which has that interesting phenomenon called blind sight, and I put the timestamps here if you guys are wanting to just go into that area of the documentary to see it. And then I have some of the vision, the lens accommodation, and also a lot of hearing, animations here. So they range from a few minutes up to like 6 or 7 minutes. So they're not particularly long, but they're really, I think, helpful in explaining how hearing and equilibrium function in the ear. And so please take the opportunity to take a look at them. Like I said, we don't have time to take a look at every single one. We'll do a few here in class. The other thing is that hopefully people saw the announcement, but I moved the in-class quiz to Wednesday. There'll be one question on the autonomic nervous system, and the rest of the questions will cover special senses up to what we've covered through today. All right? Okay. So just want to start off with taking a look at some of the anatomy of the eye. We're just about to get into the physiology. But remember, with the eye, we have the anterior part is the cornea, and then we have the aqueous humor that is underneath that. Then we have the lens. And then here, I didn't draw every structure on here, but let's say that this is the sclera. So that's a sclera. And then what's the next layer underneath the sclera? Anybody remember? The choroid, right? And so what's in the choroid? So what do we have there? So we got the choroid here. So that's got all the blood vessels in it. And it's also somewhat pigmented so that when light comes in here, it doesn't bounce around. So if it was kind of shiny like a mirror, light would come in, bounce off that shiny mirror, and bounce all around, and you would have trouble distinguishing, where that light's coming from, and your vision would be pretty difficult to discern. All right, so what's the next layer? So we've got the sclera, and then we have the choroid, and then we have what? What's the next layer underneath the choroid? Some people it can detach. Retina. Yeah, so we have the retina layer. Right, and that really is going to be like this. So it's the 3 layers. We got the sclera, the choroid, and the retina. And we have some aqueous humor, so fluid, underneath the cornea. And then we also have vitreous humor over in this area of the eye. And it's important because the right amount of pressure is pushing these things, not too strongly, but to keep a firm, pressure outward on the eye, so that the eyeball can move when the muscles are pulled, is one of the reasons. All right, so let's zoom in. So if we zoom in here, just on the retinal layer. So we're going to zoom in on the retina. Okay? We're going to zoom in on the retina. Okay, so if we do that, we'll just draw this section here. And this is retina. Okay? So this is going to be the interior of the eye, so that's going to be over here. And then this is towards the choroid. That's going to be out here. All right? Everybody with me on the orientation of this? Okay. So what we have, so let me do this. Let's draw it with the blue. So we got one cell, photoreceptor cells, that are out here. And let's call that A. So that's our That's our photoreceptor cell. And it is communicating with the next cell. We'll call that one B. And that's a bipolar cell. And the last cell that we have here it's got a long axon. It's going to be C here. That's called the ganglion cell. So the ganglion cell is the only cell of these three that can generate an action potential. Right? Okay, so that's the only one that can generate an action potential. All the rest of the cells, even though they release some form of transmitter, generate graded or local potentials. So, yeah, we're going to talk about how it's a bit backwards, okay, in just a second. But one of the things I want to point out, and you're going to want to be able to do this, is what are the structures that light passes through in order, right? So if light is coming into the eye, what's the first structure it passes through? Here, what's that one called? The cornea, right? So it goes through the cornea, and it goes through that humor, which is aqueous humor. And then this next structure that I've drawn here is what? The lens, right? So light's going to go through the lens, and then it goes through, what's this humor that's in here? Vitreous humor, okay? So if light comes along here, what's the first layer that it encounters? The retinal layer, right? And then it's going to stop at the choroid if it gets that far. But notice here, which one has our photopigments that get activated? Which one of these three cells of the retina? The photoreceptor cell. Notice that light has to pass through the ganglion, the bipolar cell, before it reaches the cell that it activates. So it's counterintuitive. A lot of times students want to flip these around because it makes sense, but that's not how it's arranged. So we have our photoreceptor cell, and our light has to pass all the way past these guys to get to those pigments. A bit counterintuitive. Now we'll circle back to this ganglion cell. It turns out the ganglion cell, its axon is really long, and it's the axons that make up the optic nerve. All right? So it turns out the ganglion cells, for all of the retina, they come out here kind of like this, and then they exit through that optic disc. And so you got a whole bunch that do this. You got some over here doing the same thing. And you can see how you generate this optic nerve here, right? So that optic nerve. Which means that this area where they exit, do we have any photoreceptor cells in that area? No. What do we have in that area? All we have are part of what cell? What part of that ganglion cell are making up that? So what's that guy? The axon, absolutely. So all these are axons exiting from the eyeball, from the retinal layer. right? So you're going to want to know the structures through which light passes in order. Okay? Does that make sense to folks? All right, so I got a slide that I'll show you guys in just a second. But here, during dark, this photoreceptor cell is actually constantly depolarized, sending out neurotransmitter to the bipolar cell. That neurotransmitter is inhibitory, meaning that the bipolar cell is constantly being inhibited. So during the dark, this photoreceptor cell is constantly putting the brakes on this system. Everybody with me so far on this? So here, if the brakes are being put on the bipolar cell, and it secretes an excitatory neurotransmitter to the ganglion cell, is the ganglion cell being activated during the dark? So the bipolar cell is constantly being inhibited? No, right? So during the dark, this is activated to release an inhibitory neurotransmitter that puts a brake on the rest of the system. Okay? When light hits the photoreceptor cell, the brakes are taken off. The photoreceptor cell no longer depolarizes, it no longer releases its inhibitory neurotransmitter, which means the bipolar cell can now release its excitatory neurotransmitter to the ganglion cell. Everybody with me on this? And so that's why I said it's a bit backwards from what we saw before. Is that usually we have the stimulation causes excitation, right? Here, the stimulation's basically pulling the brakes off of the system, and then the system goes. All right? So that's what the first few slides are that I'm going to show you guys. Any questions on this? Does that make sense to folks? Kind of how things are arranged? Counterintuitive nature of the arrangement of the cells of the retina? That light has to actually pass through the ganglion and the bipolar cell before it reaches the cell that it activates. All right. So this is pretty much what I just kind of talked to you guys about. Here we have our photoreceptor cell, and we got two types, rods and cones. Rods are involved with, say, black and white night vision, and then cones are involved with color vision. Us humans, we have 3 cones. All right, so we have red, blue, and green. So here, remember the photoreceptor cell, in the dark, it's constantly releasing inhibitory neurotransmitters onto the bipolar cell, which means that the bipolar cell's neurotransmitters, excitatory neurotransmitters, aren't being released to the ganglion cell. All right? Once light hits the photoreceptor cell, those inhibitory neurotransmitters from the photoreceptor cell are no longer being released, which means the bipolar cell can release its excitatory neurotransmitters, which can cause an action potential in the ganglion cell. And so that's pretty much what this slide and the next slide are about. Now, how many folks have heard that vitamin A is good for your eyes? Right? The reason for that is the molecule that changes shape when light hits it is a derivative of vitamin A. All right? And that's located in the photoreceptor cell. And so here, what ends up happening is you no longer get this depolarization of the photoreceptor cell when light hits it, which means that the neurotransmitter being released there, the inhibitory neurotransmitter is no longer being released from the photoreceptor cell, allowing the bipolar cell now to release its excitatory neurotransmitter, which is going to stimulate signal from the ganglion cell, right, an action potential. And remember, the axon and the ganglion cell, they exit through the optic disc, so they're pretty long. So they're carrying information to the brain for being interpreted and identified. Any questions on how the system works? Are making sense to folks? All right. So this is pretty much what I was telling you guys. So light's coming in. We have, our photoreceptor cells, our bipolar cells, and then our ganglion cells whose axons are exiting via the optic disc. And you can see that because we don't have any photoreceptor cells here, that this is a blind spot, right? Okay, so like I said, we have cones that are blue, green, and red. Some animals have a lot more. And so I'm going to kind of just show parts of this podcast that I think are interesting. Yeah, so I'm going to skip ahead in just a second, but so far, the big point is that because we have one more, cone, it gives us a multiplied number of colors that we can see. Now, there are organisms out there that have a lot more color cones than we do, which is kind of interesting. Let me zoom ahead here. All right, so that's, so here for our purposes, what we see is that for our vision, having red, green, and blue cones gives us, a good size of coverage of some of the spectrum of the electromagnetic spectrum. And the cones are more sensitive, or they give us better resolution, that's the way I should phrase it, than the rods do, but they require more light, which is why we can't see as well in the dark. We can't see colors as well. So the rods are a little bit more sensitive, but they don't give us as much resolution. All right? Okay? Does that make sense to folks? Rods and cones, and also the physiology, how it's kind of backwards with exciting the photoreceptor, in the dark, and then taking the brakes off during the light. Now, some of this stuff we've talked about before for regulating light getting into the eye, where we have the pupil, which is just that region in the iris, right? It's a hole in the iris. And there's two sets of muscles. One set of muscle is going to be involved with constricting the iris, so making the pupil diameter smaller. The other set of muscles is involved with actually, when it contracts, expanding the iris, therefore expanding or increasing the diameter of the pupil. And remember that those two muscles are innervated by two separate divisions of the autonomic nervous system. So here, the sympathetic nervous system is activated in response to what types of stimuli? Fight or flight, right? And so here, that is activating the dilator muscles, the dilator muscles contract. Yeah, that's right. And then the pupil dilates. Parasympathetic, though, is doing what type of responses to our body? So what's the responses for the parasympathetic? So rest and digest, right? And so that is going to be activating the constrictor muscle, which means that the pupil is going to become narrower. So if someone takes a poison, for instance, that's activating one of those divisions of the autonomic nervous system, how could you tell by just looking at their pupil diameter? So if they had narrow pupils, really constricted, which branch of the autonomic nervous system is that poison activated? Parasympathetic. If they took a poison that was activating the sympathetic, what diameter of pupils are you looking for? Yeah, big, yeah. So increased diameter there. All right, so some of the cranial nerves, big one, cranial nerve number 2 was the optic nerve, right? And that's starting off with the ganglion cells, right? It's a ganglion cell neuron, axons. To move the eyeball around, we have extraocular muscles that are innervated by these 3 cranial nerves, the oculomotor, the abducens, and the trochlear, right? The 3, 6, and 4 cranial nerves. Now, anybody remember what cranial nerve number 1 was? Yep, it's the olfaction, right? It's involved with olfaction, the olfactory nerve. All right, so accommodation of the lens. The lens. So you guys saw this in the lab when you're injecting to expand that kind of bladder that had water. Kind of we're mimicking the bulging of the lens. So a fattening of the lens. So here for close vision, ciliary muscles are contracted, the lens is rounded, it's bulged. For things that are a little bit further away, those muscles relax and the lens flattens. Right. So if you're looking at something up close, those muscles are staying contracted a lot, right? So how can this relate to fatigue? So think about that. Okay? So every once in a while, you probably want to take a break. Okay? So here, the reason the lens is either flattening or bulging is for reasons you see in that working model. It's changing, so the focal point of those bent light rays are coming together on the retina, right? That's the whole point here. So here for close vision, it bulges to try to get that focal point on the retina. And then here for far away vision, the lens is flattened, so that once again, changing the bending angle so that those light rays are coming in contact with the retina. Everybody with me on this? Okay. So that's called lens accommodation, and hopefully you guys spent some time in the lab to take a look at that working model. Okay. So close vision, talking about the lens bulging, distant, the lens flattens, and when it's bulging, so it's the muscles are contracted, and for distant visions, they're relaxed. The lens is kind of more flattened. Now, what about refraction disorders? We call that bending of light refraction. So here, if you're farsighted or nearsighted, what's happening? Well, it turns out that for whatever reason, the eyeball's misshapen a little bit, so that here with the light rays coming in, the focal plane's too short or it's too long. It's basically not coming together at the point of the retina. All right? So for me, I have nearsightedness, which means that without any type of corrective lenses, I can see up close pretty well, but far away, there's a problem where the focal plane, right, is going to be too short compared to where the retina is, where it should come in contact. For those that are farsighted, it's the opposite problem. The eyeball's a bit too short, meaning the focal plane is going past where the eyeball is. So how do you do the correction of this? Well, the main way is to put another lens, so you kind of pre-bend the light, right? So that now that the light that is bent is now coming at the right spot on the retina. The focal plane is now at the retina, not in front of it or behind it. Same thing with farsightedness. Right. Does that make sense to folks? Right? So basically, for glasses or contacts, what you're doing is you're putting another lens to pre-bend the light to help with focusing the light on the retina. Because without that kind of additional lens, the focal point is either before or after the retina. All right, so what are some of the steps for how we see? Well, light rays pass through the cornea, so remember to know the structures through which light passes in order. then it goes through the pupil, and then remember we have muscles that are either going to dilate or constrict the pupil. And we have accommodation that goes on, whether we have bulging or flattening of the lens. Image gets focused on the retina. All right. Light rays are absorbed by the photoreceptor cell. And so remember we have rods and cones, right? The rods are a little bit more sensitive. You need a lot more light intensity for the cones, which is why we don't see in the dark color very well in the dark. And then we have several synapses, remember, going from photoreceptor cell to bipolar cell to ganglion cell, and then ultimately the axon of the ganglion cell is going to be making up that cranial nerve number 2, the optic nerve, right? And remember, it's a ganglion cell that can generate an action potential. Does that make sense? All these series of events. Okay? So what happens after that? So here we have our axons from our ganglion cell. If we track it, they actually go through that chiasma. So remember you guys were dissecting the sheep brain? You can see this kind of crisscross. So that's what this is. So you can see for one eye it crisscrosses, and it's interpreted by the opposite side of the brain. So when we say that we have our signal and then the interpretation or the response to that signal is on the opposite side, what word did we use? Anybody remember when we talked about reflexes? So contralateral. So here the contralateral side of the brain is interpreting some of that image information, right, from the opposite eye. So here, the area that has our vision association and primary visual cortex is in the occipital lobe. So that a lot of that for the right eye is located in the left occipital lobe, and a lot of that for the left eye is located in the right occipital lobe. Now, all the signal from the eye is routed to the cortex. That's our conscious interpretation, right? Like you look at something and, oh, that's a hand. That's a cell phone. Okay? Some of this gets routed to parts of our brain that are involved with circadian rhythm. What's circadian rhythm? Sleep-wake cycle, right? Or 24-hour cycle. Turns out that light's really important for making sure that that is properly calibrated. So there's a part of our brain very deep in that some of this optic information is going to. And if you guys watch that section of Secrets of the Mind on blind sight, people can, even though they're legally blind, you put an object in front of them, they can't identify it. But if you move that object up, down, left, right, they can tell you that it's been moved up, down, left, right, which is pretty amazing. So even though they can't see, they can tell direction. Right? If you guys are interested in figuring out why that is, watch that little bit that I put out on the Secrets of the Mind. Okay? Does that make sense to folks? All right, so the processing of vision. So here, where is that main processing going into initially? The primary or association area? Primary, right? Identifying and interpreting that vision signal, right, that optic signal, is being done by association, so visual association area, right? Okay, so both of those are located in the occipital lobe. All right, so here's the visual association area, then we also have our primary visual cortex. Both of these are located in the occipital lobe. Like I said, some of the signal from the eye gets routed to some deep structures that are really important for maintaining sleep-wake cycle, circadian rhythm, and also any sensory input really gets routed through the thalamus for making sure that it gets routed to the right place, and all things that are would generate noise in the interpretation are filtered out. So both of those are in the occipital lobe there. All right. So a few disorders, we don't have time to talk about them all, but cataracts. Cataracts are clouding of the lens. So it turns out also as we age, so the lens is kind of like an onion. As we age, layers get added to the lens. And so you can think about what ends up happening as we age. Well, the lens becomes less able to accommodate. It's less able to be bulged and flattened, which is why our vision decreases as we age. All right? But cataracts is where the actual lens starts going through clouding, and so there can be actually transplants for lenses that happen. The other one is glaucoma. Glaucoma is increased ocular pressure, intraocular pressure. And if you increase intraocular pressure, you're pushing up against some sensitive structures like the retina. And you can impair, the retina, or if the pressure is too high, what it can do is it can push up against the blood vessels in the choroid. And if it pushes up against those blood vessels, maybe you don't get as much blood vessel or blood flowing to areas that need it, right? So that can be problematic. So intraocular pressure is glaucoma. Cataracts are clouding of the lens. All right, so why don't we do this? Why don't we take a 5-minute break, and after the break, we'll talk about hearing and equilibrium. All right, so we're going to move on to talk about the ear and hearing and equilibrium. They're part of the ear. And like I said, a lot of these structures, three-dimensionally, are very complicated. And so what we'll talk about is basic anatomy, mechanics of sound, and then talk about the different types of balance or equilibrium. And so here, if we look at the ear anatomy, we can divide up into 3 areas: the external ear, middle ear, and the inner ear. So the external ear has the ear canal, the auricle or pinna there, and it is going to funnel sound waves through the ear canal to this drum-like structure that's called the tympanic membrane. The tympanic membrane separates the external ear from the middle ear. The middle ear is this area here. It's a bit like the back of your throat in a way, in that it has some of that soft tissue and mucosal tissue there, right? But most of this opening, this cavity, is air. So here, the external ear, it's not plugged up with earwax, is mostly air. And then here we have air. Okay? Part of this middle ear has a opening, a tube, that goes to the nasopharynx, the back of your nose, kind of upper part of your, if you follow back to your back of your throat and just a little bit above that. This is where this tube empties. When I learned it, it was called the eustachian tube. Then it adopted another name, the auditory tube, but people were thinking that there was a confusion with this term and then the ear canal. And so it's called now the pharyngotympanic tube. So there's 3 names for the same structure. I'm sure they did that to make anatomy and physiology students have to memorize more stuff, right? So pharyngotympanic tube, though, is named for the structures on either end of this tube. So one end is leading to this tympanic cavity, where the tympanic membrane sits, and the pharynx is the other end. All right? This is the tube that quickly opens to normalize pressure on each side of the tympanic membrane. So if you go and have dramatic change in elevation, for instance, you change pressure dramatically. What ends up happening is this tube quickly opens and closes. So it's the clicking sound or popping sound you hear when you go really high up in a mountain or when you're coming down. What it's doing is that it's allowing for the pressure in the middle ear to normalize to the pressure in the external ear. Is that making sense to folks? All right. As you guys saw in the lab, this tube is fairly horizontal in children, or more horizontal in children than is in adults, which means that if there's a throat infection, it can be conducted more easily to the middle ear in children than in adults. This is why children can have earaches or ear infections more frequently than adults. This other structure, so remember also in the lab you guys saw those ossicles, the auditory ossicles, 3 really tiny, tiny bones. The malleus, the incus, and the stapes. Right? Malleus is hammer, incus is anvil, stapes is a stirrup. The reason is because it looks like a stirrup, and the malleus is connected to the tympanic membrane. You track these ossicles, the stapes here is covering what's called the oval window. It's sitting right on top of that. The oval window, and this part is all part of the inner ear. This inner ear is embedded in the temporal bone. So if you track kind of behind your ear here, kind of a bony protrusion, that's really where a lot of these structures are located. Okay? So this inner ear has some complicated structures. These are basically complicated passageways, so sometimes it's referred to as a labyrinth. And these structures are where we have the sensory structures for hearing and equilibrium. The cochlea is the structure that's involved with hearing. And for equilibrium, we have the vestibule and the semicircular canals here. Those are involved with equilibrium or balance. Now, one of the differences between the inner ear and the middle and external ear is that the inner ear, okay, on the other side of this oval window, is filled with fluid. Okay? So here the external and middle ear are basically going to be filled with air. The inner ear is filled with fluid. So if we take a look at this, zoom in on the middle ear, you can see that we have various ligaments, and also there's some small tendons and muscles that help make sure that these small bones don't get they don't have excessive force on them. And so here's our tympanic membrane, malleus, incus, and stapes, and that is going to lead to the inner ear, which is where we have the cochlea, the semicircular canals, and the vestibule. All right. So zooming in on the inner ear. So if we look at the inner ear here, we have our semicircular canals. We have our vestibule. There's 2 areas of the vestibule, utricle and seculae. I'm not so concerned you guys know those terms, but they're part of the vestibule. And then we have the cochlea here, which is going to be involved with hearing. Okay? The cochlea starts, with the oval window, and it ends with the circular window. And if you guys looked at the working model in the lab, you could see both of those. In a kind of a cross-section of the cochlea, the organ that's involved with the actual hearing that has the detector cells or sensory cells is called the organ of Corti. And it's these hair cells that are really important for sending the signal. These hair cells are mechanoreceptors, or they have mechanoreceptors on them. So if we remember olfactory neurons, what types of receptors were they? Chemoreceptors. So here, gustatory receptors. What type of receptors were they? Yeah, so the same, chemoreceptors, right? So if we talk about, say, vision, those cells had what type of receptor? Photoreceptor. And so here, now we're talking about mechanoreceptors, which means that they're activated by a physical movement, a force. Okay? And it turns out what ends up happening is this membrane, it depends upon the degree or the frequency is going to push these hair cells up against this membrane up here, the tectorial membrane, and when it does so, it causes these channels to open. All right? And so I have some videos showing this that we're going to watch in just a second. Okay? And when they open, they cause depolarization, which means that they send an action potential down these cochlear axons of these cochlear sensory cells, these inner hair cells, these hair cells, right? And then that can be interpreted in various parts of our brain. Okay. Mm-hmm. It can be, yeah, it can be, absolutely. All right, oops. All right. So that's the kind of the basics. I'm going to show you guys another video that shows how different pitches, so different sound waves, are identified in the cochlea. It's this one. So basically what we're talking about here is that if we straighten out the cochlea, right? So remember, it's kind of snail shaped. If we straighten it out, actually, let me turn this. So if we straighten out the cochlea, so that's what you guys saw in the working model in the lab. That kind of clear tube, right, was a straightened out cochlea with the membrane. And here, remember, for hearing, that we have sound waves. Frequency is how close the peaks are to each other. Amplitude is how high those peaks are, peaks and troughs. Okay? So here, higher frequency is going to have, right, peaks that are really close to each other, peaks and troughs close to each other. And then lower frequency is going to be much more extended. Now, if we look at our cochlea that we've unfurled, we basically have it so that here's our sound waves coming from the stapes pushing in that fluid, right? And it is going to go along here and then eventually along the exit towards the round window. Then we have a structure that's internal that has that's called the cochlear duct. And that has the organ of Corti. And the membrane for the organ of Corti, the basilar membrane, it actually is kind of like piano strings. So very short piano strings or strings on a harp. They're very difficult to displace. You need a lot more kind of energy to do that. Higher frequency has more energy, so it's able to displace these guys. Towards the apex here, that basilar membrane's much wider and it can be displaced much more easily, so it doesn't take as high a frequency to do that. And so you can see that higher frequency sounds are going to be stimulating closer to this oval window, and lower frequency sounds are stimulating closer to the apex. Now, each one of these hair cells as part of the organ of Corti is mapped, right, into the brain, so that if a signal is received from these hair cells, then your brain knows that, oh, it's a high frequency. From these hair cells, it's going to be a low frequency. Everybody following me on that? And you saw that in the videos. Okay? And when you guys took a look at the working model of the ear, you could see that membrane change, and you can see when you pushed in on the oval window, the round window came out in order to propagate that. All right? So here, frequency is telling us something about pitch, amplitude, how much gets deflected, right, is telling us something about decibels, or how loud it is, right? Is everybody making sense with this? Okay. So where along the cochlear duct, maximum displacement is telling you the pitch. How big that displacement at that position is telling you something about how loud that pitch is. Everybody with me on that? Because the three-dimensional structure here is complicated, and understanding how it works is also challenging. So if you rolled this thing up, right, you get the cochlea, the snail shell that we see, right? Here it's just been unrolled or unfurled. All right, so basically if we're taking a look at hearing, sound waves funneled in by the pinna or auricle, the outer part of our ear here, into the external acoustic meatus or auditory canal there. Vibrate the tympanic membrane, as you guys saw, and that is going to be transferred via the auditory ossicles to the oval window, which is moving fluid in the cochlea, and then the displacement of certain parts of the cochlea, those hairs specifically, are going to be detecting the pitch. How much is displaced in that area is detecting the amplitude, how loud it is. And then you get an action potential that's going down the cochlear branch of the vestibulocochlear nerve. So kind of back to, some folks were asking about if you can lose hearing. Well, think about, losing maybe sensitivity. So there's actually small efferent pathways that funnel into some of these to make sure that the hair cells don't overextend, or, there's fine-tuning that goes on for the pitches. And so, in some cases, those efferent pathways can be disrupted, and you can have damage being caused. There's other ways in which damage or problems with hearing can occur, too. So think about the mapping, if there is problems mapping properly. All right. So remember, the cochlear, or the vestibulocochlear nerve is cranial nerve number 8, right? And so it's this guy here. So we haven't talked about balance or equilibrium. That's going to be kind of the vestibular portion. Here we've only talked about the cochlear portion. That's involved with hearing. So you should remember that the cochlea is involved with hearing. Semicircular canals and vestibules are involved with equilibrium. All right. So this will be the last part, and we'll do equilibrium on Wednesday. So here, if we have information coming from the cochlea, it's going to be passed eventually to the auditory processing area, which is located in the, part of the temporal lobe. All right. Okay. Right. Now, we also have some processing in the midbrain for a lot of our senses. But remember, we have a primary and an association area. Primary is where we get the actual processing going on. The association is determining what type of sound. Is it music? Is it music I like? Is it music I don't like? Is it speech? Right? Can I actually understand the lyrics being sang by the singer? Right? So that's pretty complicated to be able to do that. And so remember here, in order to do that interpretation of language, spoken language is happening in the Wernicke's area, right? Identification and interpretation. Okay, so we'll leave it there. We'll pick up with looking at equilibrium on Wednesday. All right? And please take a look at those. I have other videos that I didn't show, animations. So if something was kind of fuzzy or unclear, rewatch what we watch in class, and then also take a look at some of those other ones too. All right? Great. Have a great rest of the day, and I'll see you all on Wednesday.